So, welcome everyone to the NPTEL course on food science and technology. So, this course is basically instructed by Professor H.N. Mishra, who is in the Department of Agriculture and Food Engineering, IIT Kharagpur. And I am Shikha Sharma. I am a research scholar in the Department of Chemical Engineering, IIT Guwahati. So, this course is basically this live interaction session for this course is basically for week 2. So, the assignment 2 deadline is 4th. So, please try to solve the assignment and submit on time. So, I'll be teaching this course for whole January 2026 semester. So, today we will be taking our second live interaction session. So, we'll be basically today taking the module 2, which is on food structure function relationship. So this is the module 2, which is having 5 lecture videos, including lecture 6, 7, 8, 9, and 10. So this particular module discuss about food quality characteristics, then chemical composition and the physical structure, then physical, textural, and rheological characteristics of the food, then thermal properties, and the relation of the structure of the food to its quality. So there are 5 uh, lecture videos which is discussed by Professor Mishra, and I hope you have already gone through all these 5 lecture videos. So today we'll be trying to discuss in this tutorial class about like some theoretical questions regarding these 5 lecture videos. So I hope you have already gone through these lecture videos, and I'm recording. The recording is already on for this class, so I'll be giving you all the recordings after the end of this class, and you can ask your doubts in the middle of the middle of this lecture or at the end, or also you can post your questions on Q&A group. So let's begin with today's first question, which is about the properties of food. So which of the following properties of a food involves basically interaction at the interface between two immiscible phases? So there are 4 properties like solubility, emulsification, cohesion, and gel formation. So we need to tell which of the property basically is which is of the food involves the interaction at the surface. So where the two immiscible phases, it is trying to make the like miscible phase, or it is trying to make like in trying to uh, interact at the two interface. So which of the property is related to that? Anyone would like to answer this question? Shahnaaz, emulsification. Okay. Shivaswamy, B, emulsification, and Dipti also B, emulsification. Okay, so you guys are so basically in the if we talk about the major attributes of the food, so basically they are the chemical properties of the food. Then we have the functional properties of the food products, and we have the sensory properties of the food product, and then we have physical properties and nutritional properties. So in chemical properties we have pH, acidity, chemical composition, then its redox potential. In functional properties we have different properties like water holding capacity, emulsifying property, and then solubility. In sensory properties we have around like this all taste, aroma, and how we can feel in mouth the food product, and then color, all are the sensory properties. And also about like how we can smell that food product. So next is physical property, which is like color, density, texture, specific heat, water activity. And also we can include like porosity, then volume also. Then nutritional properties. So nutritional includes macronutrients, micronutrients, glycemic index, etc. So these are basically the overview of the uh, major properties which is there in the food product. So as you all said that the property, so we are here dealing with the property related to the functional property, which is the emulsification property. So, as we all discussed that when we are missing the two immiscible phase, so the interaction is done by the emulsifiers, or the property is known as the emulsification property. So, for the functional property of a food also includes like the hydration property, uh, which includes its solubility, adhesion, and the cohesion. Then aggregation, that includes basically the gel formation and the film formation. Then interfacial property, which we are discussing here, that we are that what is happening at the interface of the two immiscible phases basically. So one is like emulsification, second is whipping, then third is forming. Then fourth property is sensorial property, which is like regarding the color, texture, and the flavor. So here we are discussing mainly about in the functional property about interfacial property and particularly the emulsification. So emulsification is the basically the functional property where the food ingredient stabilizes the dispersion of one liquid in another. Like oil in water emulsion is there. So what is happening is there is a formation of the interface between the two different layers. So emulsifiers are the compounds basically which do such function of emulsification. So what they do basically, they lowers the interfacial surface, interfacial energy at the surface, or you can say the surface tension, it will reduce. So what will going to happen? Then the two immiscible liquids, they can we can say that they can mix, or there is a formation, sorry, there is a formation of emulsion because of the emulsifiers. Then there are different type of like the emulsification system. So there are different emulsions basically. One is the oil in water emulsion, next is water in oil. Then multiple emulsions can also be there, like oil in oil, or water in oil in water. Like these are the like these are the multiple emulsions, like water, oil, then water, or oil in water in oil. So these kind of emulsions are known as multiple emulsions. Next is macro emulsions, where the emulsion particles are greater than 50 nanometers, or micro emulsions, where the particle size is around 5 to 50 nanometers. So these are the different emulsion systems. So your answer is correct that emulsification is the process or is the property of the food which involves interaction at the interface between two immiscible phases. Next question is that the emulsifiers are important because they, right now we discussed about this, so they increase the surface tension between two immiscible liquids, or they facilitate phase separation, then they lowers the surface tension and stabilize the emulsion, and fourth is increase the density of liquids. So this question is very easy. In case of milk and butter, It will be two-phase emulsions. C. I didn't understand. Can you please repeat, Shivsami? Everyone is saying C for the this question, that lowers the surface tension and stabilizes the emulsion. Okay, let's see. Yes, you're correct that emulsifiers are basically uh, they basically lowers the surface tension and like between the immiscible liquids like oil and water, because oil and water, one is hydrophilic and another is hydrophobic and hydrophilic, so it will not mix. So we need some kind of the emulsifiers or some kind of the additives to make some to decrease the surface tension, so that the emulsion whatever is making final emulsion we are getting, it will be completely stable. So that's why it is making easier for these liquids to mix and form a stable emulsion. So by positioning themselves at the interface, emulsifiers basically prevents the liquid from separation. Otherwise, if there is no emulsifier, what will happen? The water will be at the bottom and oil will be separately at the top. So they will, if we try to mix oil and water, they will completely separate, and oil will, because its density is low, so it will be at up and water will be at low. So we need emulsifiers so that we can get that stable emulsion. So the products like the mayonnaise and the salad dressings, so they need emulsifiers. So this stabilization is key to maintain the texture and the consistency on the emulsified foods. So if we see here, so this is the emulsifier. So here is the hydrophilic head and this is the lipophilic tail. So if there is oil in water emulsion is there, so what will happen? If the outer phase is aqueous, if there is beaker this and outer phase is suppose water, so hydrophilic will be outside and lipophilic will be inside, forming this kind of structure, which is micelle we say. Then another is if oil phase is outside, so what will happen? The lipophilic part will be outside and the hydrophilic part will be inside. So, like if there is oil in water emulsion, so we have two phases. One is the continuous phase, which is or if there is water in oil, or what oil in water emulsion. So there will be two phases. One is dispersed phase, another is continuous phase. So continuous phase is the one which is the in the large amount, or which is external phase, and dispersed phase will be the internal one. This is the this amount is small also, and this is dispersed in the continuous phase evenly. And multi-phase systems looks like this. So this is oil in water in oil, and this is water in oil in water. So like this, there are three systems. One system is here, second is this at boundary, and third is inside this one in both the cases. So these are the emulsions, different type of emulsions. Okay, if the question is clear, can we move to this next question? Pardon? Anyone would like to say something? In case of milk and butter, it will be a two-phase emulsion. Milk is oil in water emulsion and butter is water in oil emulsion. So you are telling us, Sivashami, the examples. Okay, make a correct pair between the instruments or the methods in the column 1, which are four methods. Carl Fisher titration, titer. Second is mass spectrometer. Third is texture analyzer. And fourth is enzyme linked immunoabsorbent assay, which is ELISA. And second column is having its use to measure the quality attributes of food. So vitamins, allergens, moisture, and chewiness. So we need to match the different methods or the instruments to the different quality attributes in the food, which it measures. Okay, let's see the answers suggested by you guys. Okay, Shiv Sami, thank you for helping us with examples. Uh, Tripti is saying B, Shiv Sami B, Gautam B, Neha B. Okay, most of us are saying B. Okay, let's see. So B is correct answer. So there are different quality evaluation methods. We have like the subjective evaluation methods in which the humans basically are the trained humans. They will do the sensory evaluation. And second is the objective evaluation, which is done basically by the instruments. So we try to mimic the properties like the for texture, sorry, for mouthfeel and for how we perceive the texture. So we use texture profile analysis or something like equipment. So this is the objective methods where we use equipments in place of human beings. So Carl Fisher method is basically Carl Fisher titled this method is basically used to measure the moisture content. Basically, it looks like this. So it has cathodes and anodes, which will going to measure even the small amount of the water. So it is basically a bipotentiometric titration method, basically. And it uses the volumetric titration to determine the trace amount of the water in the sample. So the reaction takes place is like this. So the water which is present into the uh, basically water present into the sample is basically it reacts with the SO2, which is sulfur dioxide, and it forms basically H2SO4, and H is basically it will react to iodine, so it form HI. So we will going to measure basically what is the amount of the iodine. So indirectly, uh, based on the amount of the iodine, like what will going to happen when we'll do the titration. So what will going to happen is the amount of the iodine is basically detected here. So when that we reach like the titration volume will reach, so will the color change will be there. Basic titration we have the color change, like that color change will be there. From that we'll detect the amount, we'll quantify iodine, and based on that we'll quantify how much amount of the water is present in the food sample. Second is the mass spectrometer. So basically, this is the mass spectrometer where what happens is we use it for the detailed analysis, like for identification and for the quantification of molecular structures of vitamins based on their mass to charge ratio, which is M by Z ratio. So mass spectrum, it basically plot mass spectrometer basically plot this kind of the spectrum where intensity will be there of different molecules based on the charge, like different at different retention time we'll get certain amount of the intensities we'll get for certain molecules of different m by z ratio. So what is going to happen here, like this, from here, this in this tube, what will going to happen in mass spectrometry, the sample will go. So with the, like, we can have gases like helium, argon. Basically we use helium, which is the inert gas, to carry the sample. So with sample, helium gas will go, and then what will happen, the sample will going to be heated. It will heat so high, and with that at a high heat, electron beam will be there, so it will make, from the samples we'll get ions of the sample, from the sample. So the ions will basically, they'll go the speed will be accelerated with the help of the like some kind of equipment, some kind of parts. So basically there will be the deflectors or the filters will be there. So, or magnification systems will be there. So what will happen? First, the speed will be accelerated, and after that, there will be magnetic field will be there. So because of the magnetic field, based on m by z ratio of the particular ion, it will deflect. So the deflection will be different. So some ions will fall here, some ions will fall here. So there the path will be deflected for, and they will be finally go to the detector. So the detector will detect all these ions based on its m by z ratio. So basically, if the mass is lesser, so the deflection will be higher here, and if mass will be more, the deflection will be less. So like that, and it is also based on the charge, so the deflection will be there. So this kind of uh, this is the mass spectrometer. Next is the texture analyzer. So in the texture analyzer, we measure the texture of the food product, which is basically the chewiness, gumminess, springiness, and other things we measure. Also the like firmness of the fruits and vegetables. So basically, but we'll be discussing this in more detail, and here we can see the texture analyzer, how it looks in the in the laboratory scale. So this will be the plate. It will try to, like, the force will be there. So this is the food sample. So on the food sample, force we are applying, and we are seeing when it is going to be fractured, or it will going to be break. Next is ELISA. So basically this technique is a plate like this. We have the plate based assay. There will be wells, and we'll put certain enzymes and certain like antigens, antibodies to analyze such kind of the test. So what we're going to do? So in this, basically it is used to detect and measure all the allergens, antibodies, antigens which are present in the food and biological materials. So in an ELISA, basically what will going to happen, there is antigen will be there, which is from the food. We will have some antigen. Food sample will have some antigen. So that is a target molecule, and it will be, what will do, will immobilize. Like if we see here in the image, this is the antigen we have put into the this plate, this well, we will put this antigen, and it will be immobilized on the surface of the plate. Then immobilized on the solid surface of this microplate, and then the antibody, which is coupled with the uh, reporter enzyme. So this is the antibody which is conjugated with the enzyme. So this is the antibody. So this antibody will be attached to this. So what will going to happen? the substrate will be converted into some product. So based on that, we'll get some kind of the signal or the change in the color, which we can measure using like here is the blue color conversion. So we can see different intensities. So this will be detected under UV visible UV vis spectrometer. So from there we can uh, predict or we can analyze the intensity of different colors, and based on the intensity or based on the reflectance and the transmittance, basically we can understand how much is the concentration of that color, and based on that we can understand how much is the antigen present or how much is the antibody attached to the antigen. So this is kind of the test for ELISA. So here we were there. So the Karl Fischer titer, basically it is measuring the moisture. Then MS is measuring vitamin. Then texture analyzer, basically it is measuring the chewiness. And last is the ELISA, it is measuring what how much is the allergens present. So B answer is correct here. Let's move to this question. What is an effective way to prevent browning in food products like egg powder? Rajeshwari C, Shivasami C, okay. Enzymatically oxidize glucose. Lakshmi also, Shehnaz also. So many people are saying C, which is correct also. So basically, what happens? So when egg powder is there, so it is having some glucose amount in it. So when we dry it in the maybe spray dryer, so that glucose amount is going to react with amino acids or the proteins present in the uh, egg powder, and it undergoes the Maillard reaction, which is this. So basically reducing sugar, it will react with the amino groups of the protein which is present into the egg, and it will convert into the skip base, and it undergoes it basically forms the amatory compounds of the products, and after that there will be like the reaction will go, and finally melanoids will form. These melanoids are of brown color. So because of that, there is a change in the color or browning in the egg powder. So to prevent that, we need to remove this glucose, or we need to, sorry, so we need to remove this basically glucose. So we need to, yeah, remove this glucose which is present into the egg. Or what we can do is we can oxidize this glucose so that the in the egg powder there will be no non-enzymatic browning. So if there is no non-enzymatic browning, so the color of the egg powder will remain fine, it will not change. So what we're going to do, we will do the enzymatic oxidation of glucose so that the Maillard reaction will not going to be happen, and that's why the color of the egg powder will remain fine. The next question is, which of the following statement is true? So the protein denaturation basically during the boiling of the milk does not affect the nutritional quality. Then lipid oxidation is required to maintain the quality of oil. Next is removal of phytates from the grain by milling is a desirable change during the processing. Then fourth is softening of plant tissues by heat during cooking is an undesirable reaction or undesirable change. So which of the following statement is true? The C answer, many people are saying C. The removal of phytates from grains by milling is a desirable change during processing. Okay, so you guys are correct. That basically phytates in the grains, they will going to bind with the essential minerals like the iron and the calcium. So they reduce the bio availability and the absorption in the human body. So milling is done to remove phytates so that is the desirable basically change for increasing the nutritional value in the grains so that those essential minerals like iron and calcium can be accessible to human body. But the protein denaturation during the boiling of milk, generally it affects its nutritional quality, so it is not desirable. And then lipid oxidation also it is not required to maintain the quality of oil because of the lipid peroxidation or lipid oxidation. There is the rancidity problem, so which is also not required. Next is the softening of plant tissues by heat during cooking is a desirable change. So basically softening of the plant tissues is not desirable because it degrades the texture or the firmness of the fruits or vegetables. So this is basically the structure of the phytate. So this is basically more the what is happening in this structure. If we see Fe3 plus is bind. So this is a structure of monoferric phytate where the phytic like this phytic acid is having the complex coordination bond and uh, so basically this will form with the iron Fe and it traps the iron or the essential nutrients in and this now iron will be not accessible to the human body. So at physiological pH 6 to 7, phytate has basically 6 negatively charged phosphate groups. So if we see this is a phytate structure. So here 6 basically this phosphate groups are there, charged phosphate groups are there, negatively charged. So they will bind with the metallic cation ions like the Ca2 plus or Fe3 plus or magnesium or manganese or zinc and they will after these minerals will bind to this phytate then they'll become insoluble and indigestible and they'll not be available for the nutritional purpose for human body. Okay, so far every question is clear to everyone, so we can move to this next question. In the CIE lab color space, L star represents, so it represents color intensity, lightness, hue, or saturation. D is the, everyone is saying lightness. So which is correct also, that we have in the color measurement two type of methods we can follow, colorimetry methods and the spectrophotometric methods. So color basically is very important because it affects the food aesthetics, and it is basically used also as a freshness indicator or the ripening indicator, or the quality of the food it can determine. And colorimetry basically uses three primary colors, which are red, green, and blue, which are responsible for the color perception by human eyes. But in spectrophotometer analysis, basically what it will do, it will measure the color in the full spectrum of the visible light. So what is the spectra of wavelength of visible light? Anyone would like to tell? So Shiv Sami is saying 400 to 700 nanometer, which is true also. So it will basically, in spectrophotometer, the sample will be put in the put basically, and the wavelength of the light will come, which will have the 400 to 700 nanometer. So this will be the quartz glass where the sample will be put in the spectrophotometer, and that sample will be detected in the whole visible region wavelength. So they split light basically into various wavelength and measure light transmission or reflection by the sample in the whole spectrum. So in CIE lab color evaluation system, we have three dimensions: L star, which indicates the lightness of the color. Then it is ranging, if it is 0, so it's black, and if it's 100, it's white. Then A star indicates green, red color. So if A star is positive, so it represents redness, and if A star is negative, it represents green, and B star if positive, it's yellow, and B star if negative, it's blue. So these are the values it represents. So another term is there when we measure color, that is gloss. So gloss is basically is a visual aspect of quality, which depends basically from the surface of the food, how much light is getting reflected. So that is gloss. So hue angle is basically, it is basically the qualitative attribute of the color. So it is the attribute according to which the color has been defined as a reddish or the greenish, and is basically used to define the difference in of the certain color with reference to the gray color with the same lightness. So hue basically we represent as tan inverse B star by A star. So it tells basically in the pure spectral of the color, which is from like the color wheel 0 to degree to 360 degree, which color is there from the pure spectral color. So it basically, this attribute is related to the differences in the absorbance of different wavelengths. So if there is a high hue angle, that means lesser yellow characteristics will be there. Second is the chroma, which is also known as saturation. So this is quantitative one. This hue was qualitative one, attribute of the colorfulness. So it is used to determine the different degree of difference of a hue in comparison to the gray color. So with the same lightness. So how much is the, it is different from gray color. The lightness, the higher is the chroma value, that means the higher is the color intensity, and color intensity is basically how strong or how dominant the hue will be there relative to neutral tones or gray tones of the sample. So basically chroma tells about color's purity or vividness. Like from fully saturated means, it means 100% saturated, that means no gray added, and desaturated means that it is 0% and it means that appearance of the gray. So chroma can be calculated as equals to under root of a star square plus b star square. So here if we see that the light L star relates to lightness. Is this question clear? Can we move to this question? Which among the following properties describe a material ability to withstand forces without breaking? Elasticity, hardness, strength, and flexibility. This is the mechanical property, like related to the mechanical property of the food products. If some is saying elasticity, like elasticity is elasticity. Material's ability to withstand the forces without breaking. Samriddhi, also elasticity. Okay, let's see. Answer. It is basically strength. So elasticity is basically the ability, sorry. So elasticity is basically the ability to return to the original dimensions, basically. Like rubber, if we are stretching or we are putting some kind of load or some kind of force, so after the deformation, the rubber will come to its original. If we are putting some kind of load or deformation, after that the rubber will come to its original. After deformation, it will come to the original, how it was original. So original dimensions, it will return back, right? So when basically in hardness is basically the material's resistance to the deformation of its surface due to impact, erosion, or wear. So basically hardness is if we are putting some force like impact force, so how much is the material's resistance before it deforms. So that is the hardness. And then strength is basically it tells how well a material resists deformation without failure when force is applied. So if the force is being applied on basically some metal rod, so without failure, how much it can withstand that force is strength. And this can be tensile strength, compressive, and shear strength also is a kind of strength. And then flexibility means that ability to bend easily without breaking. So it is like if it's like this, so it can bend when we are applying the force. So without breaking, how easily it can bend. So that is flexibility. So it is emphasizing the adaptability, or and also it will not return back to its original shape like in elasticity. So it is basically related to the toughness property of the material. So this question's answer is strength, not elasticity. So is this question clear to everyone that why not elasticity? Can we move to next question then? Okay. So in column 1, we have four structural textural properties which are hardness, cohesiveness, springiness, and the gumminess. And in column 2, we have different definition of these, like energy required to disintegrate a semi-solid food, then degree to which a sample deforms before rupturing, C is force required to compress a product, and D is extent to which a product returns to its original shape after compression. So we need to match here the textural properties. Shivasami is saying C here. So he is correct that the food texture is defined basically as the sensory manifestation of the structure of the food. So it encompasses the physical characteristics perceived through touch, vision, and hearing, which arises from the food structural elements and its response to the applied force. So basically, here we have different uh, texture properties. And if we see here, this is the texture profile analysis TPA. In previous slides, we have already seen the texture analyzer equipment. And here, so let's understand that the hardness basically is the force needed to deform a product to a given distance, where the force is basically required to compress between molars bite through which the incisors are there, and then compress between the tongue and the palate. So how much the force we can put. So this is mimicking these kind of the functions in the human body. So when we are chewing the food or when we are eating the food, so how much we are putting the force on the food. Second is the cohesiveness. So it basically indicates the extent to which a sample deforms without breaking when biting with the molar teeth. And third is the adhesiveness. So it is basically the force required to remove the material that basically adheres to a specific surface like lips, palates, and teeth. And next is the springiness. It measures the ability of a product to return the original shape after being compressed between the tongue and the palate or the teeth. Then is the gumminess. It is the basically energy required to disintegrate a semi-solid food to a state ready for swallowing. Then chewiness, number of the chews we are doing, like one chew per second or how many chews we can do per second. So it needed to masticate the sample to a consistency suitable for swallowing. Next is the fracturability or brittleness. So it means basically the force at the first significant break in the first positive bite area. So this is the fracturability. So this is the first positive force, maximum force we are getting here. So this is the fracturability. Whereas the overall maximum force in the positive area is hardness. The highest peak will be hardness. So here also we can see the hardness is the highest peak. Fracturability means the first peak in the positive direction. Then basically gumminess is the area 2 divided by area 1. So this is the area 2 divided by area 1. And springiness is L2, which is this L2 divided by L1. Chewiness is gumminess into L2 divided by L1. So this kind adhesiveness is the negative, how much is the negative area is there. So that is how much is the, it is adhered to our, the food is adhered to the teeth or it is adhered to the palate. So this is the textural properties. So textural properties, they are also, they comes under the mechanical properties basically also. But mechanical properties, they are basically uh, uh, they like this also. They includes elasticity, hardness, so which we also discussed in the textural properties also. So basically in mechanical property, strength is, if strength related properties are there, that means it is related to the material's ability to withstand the forces without breaking or deforming. And the deformation properties are when we are applying the force, so how much it can stretch without breaking, without deforming. And then fracture fracture related properties are when we are applying the force, so without cracking or without breaking, how much it can withstand, like fracturability, brittleness. Whereas if we are talking about deformation, it is like elasticity of the material. If we are talking about the strength, it means tensile strength, compressive strength. And there are different time dependent mechanical properties are also there, which tells over an extended period of time, if we are applying force or we are applying the cyclic loading, then these properties come into the picture, which are creep, fatigue, viscoelasticity. And then hardness and wear related properties, it is the material's strength to deformation, like hardness and the wear resistance. Then impact for related like properties, which are the respond to the sudden impacts or the forceful sudden impacts. So it is the impact, like it includes impact strength and the flexibility and the rigidity. They are also there. So it is about the material's ability to how much it can bend, and it has the resistance to bend or deformation. Then there is a term in mechanical properties like Poisson's ratio. So it is basically the material basically behave when subject to the stretching or compression force in terms of the lateral contractions or expansions. So this basically is the overview of the mechanical properties. So we were discussing on a short description of all the mechanical properties here. Let's now move to this next question, which is which of the following statement is true about Hookean solids? So Hookean solids basically it exhibit constant viscosity regardless of applied torque. Then it stores all the energy necessary to deform as potential energy. Next is the it continuously deforms under applied stress, and fourth is the it exhibits plastic deformation when stress is removed. So anyone who would like to answer this question? Okay, C is the answer from Shivaswamy side. Anyone who would like to answer this question, which has some different answers, or you have any doubt so far, you can also tell me right now. Okay, let's see this question. So the answer is B, which is Hookean solids store all the energy necessary to deform as potential energy. So Hookean solids basically and the Newtonian fluids have this linear relationship between the applied torque. So basically what is happening in this? They have basically constant viscosity regardless of the applied torque. So if we talk about In the rheology, fluids are basically having the viscous plus elastic both nature. So viscoelastic behavior we say. So basically they store energy through the elastic component and dissipates some energy through the viscous component. So Hookean are in the elastic, like if we do the rheology, there will be deformation. In deformation, there are two classification, elastic, non-elastic. So Hookeans and non-Hookeans are under based on the classification based on the elastic nature. And in elastic, there are viscoelastic and viscoplastic. Whereas when we talk about the flow, so they can be viscous or plastic. In plastic, Bingham, non-Bingham, and in viscous, we can classify Newtonian, non-Newtonian. So this is the basics of the flow behaviors and the deformations. In the elastic materials, Hookean solids are basically the one which have the elastic behavior, and they return basically to the original shape once the applied stress is removed. So if it basically they follow the Hooke's rule. So what Hooke's rule say is basically F equals to minus kx. So it means that when we are applying some kind of the stress, so if we are applying some stress, so strain is directly proportional to stress. Like this, if we see strain stress curve, so strain is directly proportional to stress is there. So this Hooke's law is followed basically by the Hookean solids. So here basically the deformation will be in the elastic limit. It so it can return to the original shape. So they do not deform permanently or exhibit viscosity. So it does not have viscous behavior because it doesn't dissipate energy. Instead, they store the energy from the deformation as potential energy, which is fully recoverable when the stress is released. So when the stress is released, whatever the potential energy is stored, it can be recovered. So this behavior is consistent with the Hooke's law. So which means that the stress and strain are within the elastic limit are the proportional. So they follow the Hooke's rule. So Hookean solids are the one which stores all the energy in the form of the potential energy. In basically when we apply the Hooke's rule, so we have different terms also modulus. So when there is a stress to strain ratio, so we know it as a modulus basically. So that if we say strain to stress ratio, if we are reversing, so that is compliance. We say it as a compliance. So modulus can be basically Young's modulus, shear modulus, or the bulk modulus, three type of modulus. So Young's modulus is also known as modulus of elasticity. So it defines as the ratio of the normal stress to the normal compressive strain or the tensile strain. Whereas the shear modulus is the modulus of the rigidity. So it is a relationship between the shear stress by shear strain. Bulk modulus is the force is applied from all the direction which results in the volume change. So it is the bulk modulus. So this is the modulus. So this modulus can vary depending on what kind of force we are applying. So is this question clear to everyone? Can we move to next question? Oscillatory tests are used to measure The blank behavior of foods and biomaterials, elastic, plastic, viscoelastic, and Newtonian. So now we are discussing the oscillatory tests. So there are two kind of when we have some food. So when we are doing its rheological analysis, so there are two type of the uh, test we will do. One is the rotational test, and another is the oscillatory test. So we are talking right now about the oscillatory test. So in rotational test, what we do is we get the flow curve, or we measure the viscosity of a material. Whereas in oscillatory test, we do amplitude sweep and the frequency sweep. So everyone is saying C, viscoelastic behavior, which is true also. So as we discussed that the rheological properties we measure using the rotational test and the oscillatory test. In the oscillatory test, its amplitude and frequency sweep, and rotational test flow or viscosity curve, which is a function of shear rate or shear stress. So this is the basically this and this is the flow curve. So here shear stress by shear strain will be there. So which kind of behavior that particular fluid is behaving, or that particular viscoelastic property, like that material is behaving, and we can plot it in terms of viscosity versus shear rate also. So this is also so. So this is a flow curve which is under the rotational tests. So these are basically the behavior of these two graphs are telling basically the behavior of the food. It can be Newtonian, it can be non-Newtonian. It can be basically shear thinning, shear thickening, or Bingham plastic, or Hershey Buckley, or these are basically the time independent fluid behaviors, time independent. And this one is the time dependent, which is thixotropic and rheoplastic. So with respect to viscosity, the time is with the time time like with the time is measured with time, how much is the viscosity varying. So if the viscosity is decreasing with time, it's thixotropic fluids, and if it's increasing, it's rheopectic fluids. So these fluids, they basically can be measured using these are the rheological properties of a material which can be measured using rotational tests or oscillatory tests. So oscillatory test is basically, it characterizes the material's viscoelastic and deformation behavior. It shows how they store energy and dissipates the energy. So it is basically having different viscoelastic modulus. So which is the storage modulus G prime, loss modulus G double prime, and the complex modulus is G star. So this is the in the oscillatory property. So if we see here, the oscillatory test basically they use to measure the viscoelastic nature of the food product, where it measures both the viscous component and the elastic component, which means it measures the G prime and G double prime of a material. If we talk about the elastic, plastic nature and the Newtonian, so Newtonian is this one. So oscillatory test is like basically if we talk about the Newtonian, it means that with the shear stress and shear rate, it is forming the straight line, which is directly proportional. In the plastic, it means after a particular yield stress, we are getting the same kind of like stress versus shear rate. It's directly proportional, like in Bingham plastic. But after a particular yield stress, if we see the pure elastic component, so that means when we are applying the stress, it is under stress, and when we are applying the shear stress is not there, it is in the original form. So this is the elastic component. Then viscous component is when we are applying the stress, then it is under some strain, but when we are offing that stress, it like that particular strain will be continued. So in viscoelastic property of a material, when we are applying the stress, shear stress we are applying, so shear strain will increase first, and then when we are offing the shear stress, so what will happen, it will try to uh, like try to get the original form to somewhat, but this part after certain time it will not get, like the strain will be there in the product, it will not completely come to the original form, so that particular energy is basically already dissipated, so it will not come to the original form. So this kind of, this much strain will be always will be there in the product. So this is the viscoelastic kind of property. Okay, so let's move to this question. The storage modulus, which is G prime, in the oscillatory test represents viscous behavior, elastic behavior, phase change behavior, or thermal expansion. Can you give example of each shift? I mean, which example like do you want? For Newtonian, non-Newtonian, like that? So toothpaste is a we have this ketchups and tooth. Yes, this graph. Okay. So basically, Newtonian are the water is a basically Newtonian fluid. So it is it will it when we will apply the stress strain, the like the it will form the same amount of the there, like the say when we'll apply some stress, the same amount of the strain will get. Okay, and then shear thickening, we have the cornstarch solution. So if the cornstarch solution is there, so when we'll apply the stress, initially the strain will not be like that much. After certain point of time, the cornstarch solution will become thick. So that is shear thickening behavior of the material. The viscosity will change. The and then shear thinning behavior we can see in the basically blood. So what will happen when we'll apply the stress? So it will start thickening after uh, eh, after certain stress when we'll apply, it will start thickening. Initially it will not be thin, sorry, it will be thinning. So it will not be thin, but after that it will thin. And then if you talk about the Bingham plastic toothpaste, if you see initially if you like not certainly toothpaste, but you can say toothpaste. So what happened? So when you apply initially yield stress, so it will not initially come out of that tube. So after you apply the stress, it will it will change its behavior. Its viscosity will change. So that is yield stress. After particular stress, the viscosity change and it will start behaving like another fluid. Initially it was another, then like it was not that thin, but after we apply in ketchup also, initially if we apply the stress, uh, then what will happen? The ketchup will be soft, like its viscosity will decrease, it will be thin, and then it can come out of the bottle. There are different terms in both graphs. Yeah, because one first graph, the first graph is a time independent, like when we are not taking time into the consideration, then the relation between stress and strain will give certain kind of behaviors of the fluid that is depicted by first graph, and second is a time dependent behavior. So when it here time is into the picture, so with respect to time, how much is the viscosity changing? So that's why these two are the different graphs. Is it clear, Shivaswamy? Can we move to next question? Okay, Shivaswamy, can I give you your the detail with the detailed fluids, like which are the different examples? Can I post on the Q&A group for each type of the fluids? I can give like 4 to 5 examples. So I will make one table and where you will have different kind of the fluids and the different examples for it. Okay, is it fine? Can we move to this next question now? Anyone who would answer this question? Okay, Shiv Sami, thank you for your questions. Anyone who would like to answer this question? Shiv Sami, A, viscous behavior. Shehnaz is saying elastic behavior. Okay, let's see this one. So the elastic behavior is correct. So the storage modulus, which is G prime in the oscillatory test, it represents elastic behavior of a material, basically where the energy is stored and recovered per cycle of the deformation, and reflecting how basically how well a material can return to its original shape after stress is applied. So G prime is basically crucial for understanding the solid-like behavior or the elastic properties of the food materials, like such as the gel or for the dough or for the polymers also, and is used to evaluate the structural integrity and firmness of the food matrix, whereas G double prime tells viscous behavior. So it is a G double prime is a loss modulus, and it tells about the energy dissipated in each cycle. And this is the phase angle, which is also the, we say, the tangent of the phase angle. It describes the elastic, which is G prime to G double prime relation, it tells. So if 10, this tangent is less than 1, so it is elastic behavior is dominating. And if 10 is greater than 1, then the viscous behavior is there. So 10 lost this 10 equals to, so this is G double prime by G prime. So if its value is greater than 1, that means G double prime value is greater, so viscous behavior. So if its value is less than 1, then that means G prime value is more than G double prime, that means elastic behavior dominates. So in when we discuss about the oscillation behavior, there are two different sweeps, amplitude sweep and the frequency sweep, which has already been discussed in the lecture slides. So I'll be just quickly revising it. That amplitude sweep, basically it has the controlled strain or stress will apply to a material to measure the response. So it's from this we get to know the material's linear viscoelastic region where the material behaves elastically, and after that particular critical stress or strain, the material will start deform irreversibly. So basically this is the property of a material which is basically viscoelastic. So and when we do the viscoelastic behavior, study the viscoelastic behavior of suppose polymers or some kind of dough. So what will going to happen when we are applying the stress, when we are applying some kind of like stress, or this basically this amplitude sweep is a measurement between shear rate here G double prime, or here can be stress also, shear stress also. So there will be this alveolar region. So in alveolar region, what will happen? The material will be fine, it will not deform. But after this critical strain or stress, what will going to happen? This critical point, the material will behave as a viscous material, or it will deform completely, it will deform and it will not going to like it is a irreversible deformation after this alveolar region. So from this amplitude sweep measurement, we can define a material, how much that particular material, which is the viscoelastic material, or which is like a polymer or a dough, how much it, what is the reason of its alveolar. So how much is the, we can apply the stress so that it can come back to its original or reversible phase. So here what happened in rheometer, we put in like this can be parallel plate or common plate. So here will be our material, and we can apply oscillatory forces like so. This will move both sides, it will oscillate, and we can get what is the effect on our food product here. Next is the frequency sweep. So here we apply basically some kind of frequency, oscillatory frequency, which is omega, to the sample. So we can see how much is the deformation there. So from there we can understand what is the property of the material, like the viscoelastic property of the material at different frequency. Sorry. So with the change in the frequency omega, we try to understand here basically the material's viscoelastic behavior, or how much it is able to deform, or what is the internal property of or internal structure of the material. From this, we can understand whether a particular product is a gel or is basically emulsion also sometimes. But basically, it is a gel or not gel. We can understand if it's more like a solid or more like a viscous, or it's more like a elastic. It shows more like elastic property. Or it should, from this also, this we can predict also whether it is basically a shear thinning material or shear thickening material also. So here in this frequency sweep, we get such curves like G double dash with respect to frequency. So it can cross, it can go like this, it can go like this. So in this, if we see G prime is greater than G double prime. So that means what is happening? Elastic nature of the material is more than the viscous nature. And in crossover, we can see before the viscous nature was more, but after that, elastic nature of the material is more. So like that, we can predict. So if the previous question is clear to everyone, can we move to this next question? Which of the following statement with respect to the boiling point evaluation of liquid is are correct? So boiling point elevation, we know that we have the different colloidal properties. So we have four properties basically: elevation in boiling point, depression, freezing point, like that. We have four, right? So it is talking about boiling point elevation occurs when a solute is added to a solvent, raising the temperature at which the solvent boils. This is statement one. And second is amount of the boiling point elevation is basically independent of concentration of the solute. So which statement is basically we need to tell correct. Swami is saying A. Okay, so A is correct here, only one. That boiling point basically of a substance is the temperature when like when we say when that the particular material is having a particular boiling point, that when the temperature is such that its vapor pressure will be equal to the surrounding your atmospheric vapor pressure. So that means there is a phase change from liquid to gas. That means it is boiling. So boiling point. Vapor pressure of most aqueous solids, basically it is lower than that of the water at the same temperature. Therefore, given for a given pressure, boiling temperature or boiling point of a solution can be like higher than that of a pure water. So boiling point is basically useful in evaporation and drying operations. The increase in boiling point of liquid foods is a property of basically interest when we are designing the some kind of evaporators and the dryers. So boiling point elevation basically occurs when a non-volatile solute we are adding to the solvent, so which raises the boiling point of the solvent. So we can see from this picture, when we are boiling pure water at 100 degrees Celsius. So this is the molecules of the water, and the vapor pressure of the surrounding, it is equal, it is at equilibrium. So we are saying that it is a boiling point of water, and when we are adding some non-volatile solutes here, like we are adding sugar or we are adding salt or some other thing, other solute. So what will happen? It will occupy some of the spaces at the surface. So because of that, boiling point of the this solvent, which is water, is basically elevated. Now we need the more temperature to boil that particular solvent, which is in the now in the form of the solution. So this effect happens because the solute particles disrupt the formation of the vapor bubbles, so require more energy and a higher temperature from the liquid to boil. The extent of boiling point elevation basically depends on the solute's concentration and the solvent's property, and thus the boiling point change is dependent of the concentration. So we can see in this graph that this solvent, which is pure, it's uh, basically when we are seeing its uh, here, its temperature of boiling, it is somewhat here. But when we are making its solution, at particular pressure, we are checking this. So when we see after the making the solution, its boiling point elevates, temperature elevates. So the vapor pressure curve, this is the vapor pressure curve for the solution. So it lies below the curve of the pure water. That means this is the temperature increase. So if we see the difference, that is the elevation in the boiling point. So this boiling point elevation is basically, it is proportional to m, which is how much we are adding the, this is the m is the molality, basically molality of that particular solution. So if we remove this proportionality sign, it will be equal, and kb is basically the molar elevation, or it is basically the boiling point, or basically it is a constant which is denoting like uh, about the boiling point. And this del TB is basically the difference of both the boiling point when it's in the pure liquid, which is in the solvent, and solution, which is solute plus pure liquid. And also there is this formula to calculate, to do some, to solve numericals. So here del TB is equal to 1000 into kb into W, which is the weight of the solute, divided by m, molar mass of the solute, into W, which is the weight of the solvent in grams. So we can calculate del TB. Okay, let's move to this next question. Which of the following statement is true with respect to latent heat in the biomaterials? So latent heat is basically it involves heat exchange without a temperature change. It only occurs when the substance is transition from solid to liquid. It measures the heat energy required to increase the temperature, or it only occurs when the substance transition from solid to liquid. Anyone who would like to tell what is latent heat of biomaterials? So C, Shivaswamy is saying A, and C is okay. So it measures the heat required to increase the temperature. Latent heat of biomaterials. So there are specific heat and latent heat. Pradeep is A. It involves heat exchange without a temperature change. Okay, let's see the answer. So it's basically, it involves, Pradeep is correct. So it involves heat exchange without a temperature change. Basically, latent heat is the one which means that energy is absorbed or released during the phase change. So phase is changing, like if something is in solid, which is in like ice is there. So ice is converted into water, which is okay. So that means the phase is changing here without changing of its temperature. So ice can be at particular temperature and the temperature will be same, like 0 degree to 0 degree can be there, but the phase is changed from ice to liquid, from solid to liquid. The state is changed. That is latent heat. When we are talking about the heat, which is product heat, that means if a product is in liquid 20 degree and it is heated up to 40 degree, then we are taking specific heat. So it can be used in the processes like freezing and drying, where we can change the, like we can have this latent heat. So it can be expressed in the joule per kg. Its unit is joule per kg. So heat exchange during phase change from liquid to solid is heat of fusion. So this is latent heat, and heat of solidification also we say. So if we are freezing water into ice or we are thawing it, that means the temperature is basically same, but the state is changing, either solid to liquid or liquid to solid. So latent heat of vaporization also is the latent heat. So here energy is changing from the liquid to vapor, like drying or evaporation process. So water is converted into the vapor. So that is also latent heat. So phase is changing, where a liquid is vaporized for ease of separation from the mixture. So if we see the diagram, so when we are supplying the heat, this solid is converted into liquid, and if the like if it's at the same temperature, that is latent heat of fusion. When the liquid is converted into gas, that is latent heat of vaporization. So Q equals to ml. Q is heat absorbed or released, m is mass of the material, and L is latent heat capacity. Whereas when we discuss the specific heat, that is mcp delta T. Q equals to mcp delta T. This is specific heat. It is change in temperature. So if it's from 20 degree to 40 degree, or like that, temperature is changing. So we use specific heat, not latent heat there. Next is, okay, next question we discuss. Heat transfer during food processing can occur through conduction, convection, or radiation. Anyone who would like to answer this question? Okay, D, Shivaswamy, Rajeshwari, both are D, conduction, convection, and radiation. Okay, let's see. So you guys are both correct. So heat transfer in the food processing, it occurs in the conduction, which means solid heat transfer, convection fluid heat transfer, and radiation is electromagnetic wave transfer. And this mechanism can be alone or can be simultaneous. So if we are boiling the water, we can see from this solid metal, that is conduction, then in fluid it is going, then it is convection, and the radiation from the warm or the heated metal. Okay, let's see this next question. Which statement is true about heat transfers in food? Statement 1, all heat transfer modes are dependent on temperature difference. Then convection is basically heat transfer by fluid movement, and conduction only in liquids. Swami, see, both 1 and 2 statement are correct. Let's discuss this question. He is correct that both 1 and 2 are correct. So heat transfer basically only occurs with the temperature gradient, area of higher temperature to lower temperature. That is the second law of thermodynamics. So heat transfer, so statement 1 is correct, that dependent on temperature. Then conductive heat transfer basically is there where heat transfer is there and there is a transfer of energy which takes place at the molecular level. Heat transfer basically takes place in solid and it is the movement of the heat by direct transfer of the molecular energy with the solid. So there are two theories regarding it. So one is that the molecules of a solid material, it attains additional thermal energy and they become more energetic and they'll vibrate, which we can see in image, that there will be heat. Suppose this is a metal rod. So when it get heat, the molecules will vibrate and the uh, energy will transfer from one particle to another. So, so more energy and vibrate with increased amplitude of vibration while confined in their lattice. So they will be at their original place, but they will vibrate and transfer the vibration and the energy to another molecule. So these vibrations will be basically transmitted from one molecule to another without actual translatory motion of the molecules. Actual motion will not be there, but the vibration will transfer. So heat is thus conducted from the region where high temperature is there to the lower temperature. This is the one theory. That's how temperature from higher temperature to lower temperature heat transfers, like this, that at higher temperature particles will get some energy, they will vibrate and transfer the vibration to the at the lower temperature. That's why heat transfers. Second, and but they'll all the molecules will be at their particular position. Second statement says that the conduction occur at a molecular level due to the drift of free electrons. The second theory says that conduction takes place due to the free electrons motion. Free electrons will be there and they'll move. So these free electrons basically are prevalent in metals. So metal has high conductivity and they carry the thermal and the electrical energy. For this reason, the metals are basically good conductors of like the silver or the copper. They are very good conductors of the electricity as well as the thermal energy. So the conduction only occurs in liquid, that is wrong. In solids, which is correct. So next is convective heat transfer. So when a fluid or the liquid, which is fluid means liquid or gases, it comes into contact with a solid body, such as the surface of the wall. So if the wall of this container is heated and fluid is coming, which is liquid is coming in contact with that, that is convection. So heat exchange will be there between the solid and the fluid whenever there is a temperature difference between the two, with the help of the circulation or the motion of the molecules. Then heat transfer always they occur by group of molecules. Transfer will be there and from the because of the gradient of density or agitation. So what will going to happen? The fluid particles which is at the bottom, they will heat up and they will come at the top, and the cooled fluid particles, they will come at the bottom. So cooler liquid particles will sink down and the warmer will come up, which is they will rise. So that's why there is a circulation motion or circulation of the fluid, and that's why the energy is transferred in the whole container or the whole in the whole fluids. So during heat and heating and cooling of the gases and the liquids, the fluid streams exchange heat with the solid surface by convection, depending on whether the flow of the fluid is artificially induced or natural. So if the uh, this convection motion is a forced one, like we are putting some kind of the fan and we are doing, then that is a forced convection. And if it's doing like in this here in container, if the motion of the particle is natural, so we can call it free convection. So forced convection involves some kind of mechanical means like pumps or fans to induce the movement of the particles of the fluid. Whereas in free convection, due to the density difference of the warm and the cold particles, due to the temperature gradient between warm and the cold particle within the system, there will be the motion because of density difference. So both these mechanisms can result into laminar or turbulent flow within the fluid. So basically, if there will be turbulent flow occur, so mostly in the forced convections of heat transfer, the turbulent flow we can see. So turbulent flow are the one which has higher Reynolds number, whereas if the Reynolds number is less than 2000, it's laminar flow. And Reynolds number is inertial force by viscous force. And also there is one transition flow which is 2000 to 4000 is transition and greater than 4000 is turbulent. If Reynolds number, I'm talking about Reynolds number right now. So the second statement, which is convection is the transfer of heat by the fluid movement, is also correct here. Third mode of the heat transfer is radiation heat transfer, which occurs between the two surfaces by emission and or basically one will emit and another will absorb the electromagnetic waves or the photons. So it requires no physical medium for its propagation. It can even work in the vacuum. So moving at the speed of light like the solar radiation, which is a natural form of the radiation heat transfer. So the liquids are basically the strong absorbers of radiation. Gases are transparent to the radiation, except the ozone one, which basically absorbs ultraviolet radiations. And the solids basically they are opaque to the thermal radiations. Therefore, when we are dealing with the radiation, so solid materials such as the solid foods, so we will deal with the surface of the material because these are opaque. The foods are the solid materials and they are opaque to the radiations. So when we talk about the microwave or the radio frequency radiations, they will penetrate into the food, but not the thermal radiations. So all objects at temperature greater than 0, they will emit certain kind of the thermal radiations. So the heat we can calculate Q equals to Stephen Boltzmann constant sigma, which is constant value, then into E, which is emissivity, and then into A, which is area, where this uh, surface we are using, into T to the power 4, which is temperature to the power 4. E will be equals to 1 for a perfect black body. So this diagram tells that how the food is being cooked with the help of the radiations. So radiations will come cook the surface of the food. So one is this particular part is emitting the radiation and food is now in the absorption part of the radiation. So these are the three type of the mode of the heat transfer. Let's move to this next question about Fourier's law of heat conduction. So it states basically the rate of heat transferred through a material is proportional to the blank. So what is Fourier's law of heat conduction? Anyone who knows about it? Shivaswamy D. Square root of temperature difference. Okay, let's see this solution. So the temperature difference and the area. So Fourier's law of heat conduction is basically state that the heat rate of heat transfer, which is Q, is directly proportional to the surface area. This is the surface area from which the heat is transferred. So this is the surface area, this one. Uh, and and also it is directly proportional to the temperature gradient, which is temperature difference from the outside to inside. What is the temperature difference is there? So it is directly proportional, and it is the it is inversely proportional to the wall thickness, the thickness of this wall. So this is Fourier's law of heat when we are doing the conduction. So heat conduction, that it is the proportional to the temperature difference and the area, but it is inversely proportional to the wall thickness. So mathematically we write it as minus k, q equals to minus k delta t by delta x into a. So here we are taking basically, this is in the minus gradient, minus t dt by dx. So if we talk about the second law of thermodynamics, so heat will always conduct from the higher temperature basically to the lower temperature. So higher is T2 and T1 is at the lower. So from higher to lower temperature, heat flows. But if we talk about the distance, this is x2, which is greater than x1. So if we take the gradient dt by dx, it will be in minus. So the gradient dt by dx is negative because the temperature decreases with increasing in the value of x. Therefore, a negative sign is used to obtain the positive value for heat flow in the direction of decreasing temperature. So this is the Fourier's law of heat conduction. Let's discuss Newton's law of cooling, which applies to heat transfer from a solid to the surrounding, which solid, gas, fluid or liquid. Anyone would like to answer this question? Newton's law of cooling. Devaswamy D. Solid to liquid. Basically, it is both liquid and gases, so it is fluid. So it basically Newton's law of cooling, it applies in the convective form of the heat transfer, which is between the solid to the surrounding fluid, which is the air or water. So here what happens, the equation is Q is directly proportional to area and temperature gradient. So whatever is the temperature gradient between the surface and T infinity is the air temperature. So T as surface temperature minus air temperature, which is at infinity location. So we are writing T infinity here. So heat transfer is equals to H, which is the convective heat coefficient, and area, which is the this area. So it depends on the area. So Because of the viscous property of the fluids, a velocity profile is set up within the flowing fluid, with the fluid velocity decreasing at zero at the solid surface. So we are saying that since we are saying that the fluids are having this viscous property, so the velocity profile is not the same. It will be zero basically at the solid surface. So here at the solid surface, the velocity of the air is zero. So here the temperature is the same as Ts, the temperature, which is the surface temperature. So overall heat transfer will be basically from this solid surface. Can you please mute yourself? Okay, from the solid surface to the fluid, flowing fluid is proportional to basically the surface area of the solid, which is having the area A in contact with the fluid, and the difference between the temperature, which is temperature of the surface to the air. So that is the, and here H is convective heat transfer coefficient. So it also depends on the which medium we are, which is dependent on the medium or the fluid we are taking, the properties of that. So if we are taking air, the H will be of air. So this basically law is used to model the cooling of the objects in environment where a fluid medium facilitates the transfer of heat, not for the radiation or not for the conduction in a vacuum. So the convective heat transfer coefficient H basically is not a property of the solid material. This coefficient, however, depends on number of the properties of the basically the fluid, the density, specific heat, viscosity, and the thermal conductivity, or the velocity of the fluid, geometry and roughness of the surface of the solid object in contact with the fluid. So H depends on all these parameters. And if we see here, it is depending on, we can see fluid property. For air, it's different. And for condensing vapor, it's different. So here, answer is fluid. Okay, let's see this question. Food structure refers to the organization and binding of dash structure element in a unit, macroscopic, similar or different, chemical and artificial. Shivasami is saying C, chemical. Rajeshwari is A, macroscopic. Okay, let's see the answer. So it's similar or different. So the food structure, basically, it describes how various elements, they are basically, they can be similar or they can be different, and they are organized and interconnected with each other in the food. So these elements can range from like proteins, polysaccharides, fats, which can have different size and shapes, which contribute to the overall property of the food. So the type of structures can be fibrous, which is in meat, fleshy materials like fruits and vegetables, encapsulated embryos like grains and pulses, and complex fluids like milk. And the principal structural element which is present naturally in the food products are like cell walls, starch granules, protein bodies, lipid bodies, cells, and tissues. They are the naturally present structural elements in the food. So the natural food structures, we can see that this is the micro structure level. We can see that this is a fleshy structure where the hydrated cells that are bound together at the cell wall, middle lamella, they exhibit the turgor pressure, and here encapsulated embryos are there. So this is the protein, this is the cell wall. Inside that protein is there, oil bodies are there. So dispersion of starch, proteins, lipids into the discrete pockets. So these are the basically examples of how the natural food for the food in foods, natural structural elements inside that food. If we do FESAM images of that food product, we can see these kind of the structures. Next is match the structural hierarchical levels, which is the sub-molecular level, macromolecular level, colloidal level, and macroscopic level to the description, which is aggregation of macromolecules into gels, visible structures like tissues and cells. Third is interaction between the functional groups. Fourth is changes in the shapes and the size of the polymers. So we need to match the structural hierarchical levels with its descriptions. Anyone who would like to say anything? Okay, Shiv Sami is D. This one. C is the correct answer here. So let's understand why. So the size of the junction zones or the regions, the junction zone regions, where the segments of two or more macromolecules are joined together, the nature and the strength of the connected. So the size of junction zones, regions where the segments of two or more junction zones are the regions where the regions where the segments of of two or more macromolecules are joined together. So when the two or more macromolecules are joined together, that are the junction joints. So their size, the nature of the and the strength of these intermolecular forces, they define the structural hierarchy. And there are different substances like proteins, carbohydrates, fats, water, they all will be there in the food product and they'll make certain structure of the food product. So the structural hierarchy of the food, it ranges from the smallest to largest for the biopolymer interactions. So there are sub-macromolecules, macromolecules, sub-supermolecules, and macroscopic structures. So submolecular levels, basically we see it involves the interaction between the functional groups such as the chemical bondings that influence the stability of biopolymers or enzymatic modification that changes the structural function of the biopolymer, and it corresponds to the size of the monomers. Second is the macromolecular levels. It describes the change in the conformation and the size of the shape of the large molecules like the proteins and the polysaccharides, and it correspond to the size of the macromolecules. Colloids level, it refers to the aggregation of these macromolecules like gels or the emulsions that affects the texture. So it corresponds to the colloidal dimensions and greater. Lastly is the macroscopic levels. So encompasses structures visible to the naked eyes. So such as tissues or the cell organization, which we can see from the naked eyes, it impacts the physical appearance and the texture of the food. So if we see here, submolecular level, it is basically the interaction of the functional groups. Then second, macromolecules, it is the it changes in the size and shape of the polymers. Then at the colloid level, aggregation of the macromolecules into the gels, and macroscopic level is visible structure which we can see like tissues or the cell structures. Okay, let's move to this question now. higher porosity in food structures can result in reduced flavor intensity, lower the shelf life, decreased moisture content, and enhanced flavor release during mastication. Anyone who would like to answer this question? Sivishwami, Rajeshwari, C. Decrease the moisture content. Higher porosity, decrease the moisture content. Okay, let's see. So first understand what is porosity. So we can see in the breads like this structures of air in sponge cakes, in popcorns, in puffed cereals we see. So all these food materials, they have some kind of porosity or the air spaces in them. These internal pores, they can be closed pores, they can be basically blind pores, and they can be throw pores. So closed pores are the one which are completely closed from all the sides, which we can see in this image. Then there are blind pores where they will be closed from the one side, and the throw pores, they will be open from both the sides. So the porosity of the material, they can be measured using the air comparison, or we can do from the air. We can fill air like the helium, which is the inert one. So we can fill and we can measure through the cyclometer, and also from the porosimeter we can calculate based on the liquid extraction method, and we can also calculate from the image analysis like the FESM we can do to find the porosity. And it here in this question the answer is B, not C. So let me explain why. So higher porosity will be there in the food structure. That means the surface which is exposed is more. So what happens? It will facilitate the release of the flavor compounds during the mastication. So when there is a porous structure is there, so there will be easily breakdown and the quicker release of the flavors from the food product that enhances the sensory experiences in the mouth. Whereas we can also see that the porosity it basically affects the moisture retention and shelf life of the product also. Like if the higher the porosity will be there, so the more microbial attack will be there, and the lower will be the shelf life, or higher will be the porosity, that if we are drying, the moisture content will be low, but it maximum like it is affecting to the flavor. So the most direct effect on flavor perception is there during eating. So it will intensify the release of the flavor. It will not reduce, but it will intensify, enhance the flavor. So uniform, if there is a uniform pore structure is there, so it basically protect the distraction during the shrinkage when we are drying the food product. Also, it enhances the rehydration property. It increases the surface area also, and porosity, if there is highly porous or the porosity also, it affects the thermal properties like thermal conductivity or the diffusivity, and also like the mechanical properties as well, like the texture of the product. And the thermal conductivity basically depends on the porosity, pore size, shape, its distribution, how much is the void there. uh, Void means empty space, and how what is the arrangement of these voids, and what is filled in this void, like air is filled or the water, and if it's homogeneous or not, if the structure is homogeneous or not. So all these will going to affect the food properties. All these by the porosity or the void spaces, they will going to finally affect the different food properties at large. So is this question clear to everyone? So we already discussed today the basically the food properties or the food quality characteristics we discussed about food quality attributes, the chemical composition, the physical structure of the food, the desirable and undesirable attributes in the food, and how we can measure. Maybe we can measure using the Carl Fisher method, like the moisture, and from like texture, we can measure the texture analyzer, or the rheometers, we can measure the rheological properties, like that. And then we discussed about the physical properties like porosity, color, like L star, A star, B star value, then boiling point, elevation in boiling point, and the surface properties like the emulsification and the emulsifiers we discussed. Then we discussed about the textural and the rheological properties like the viscosity, flow curves, amplitude sweep, frequency sweep we discussed. And then we discussed about the also the texture profile like chewing, springiness, gumminess also we discussed. Then we discussed about the thermal properties like the heat transfer, the conduction, convection, radiation. And then we discussed finally about the food structures, the and how the physical properties like the porosity affect the food compositions also affect the structures and its quality. So in the next week, we'll be discussing about major chemicals and biochemical reactions in the food, oxidative reactions, hydrolytic reactions, and what are the different factors affecting the chemical change in the food and the enzymatic processes in the food. So I'll be uploading the video of this lecture on my YouTube channel, and PPT will be on this drive. And if you have any doubt, please paste your questions on Q&A group. Also, I'll be pasting one table regarding the different food products and whether what is their behavior, like the rheological behavior of these food products. I'll paste. So please read. I hope this week I have answered all the questions. This week's question, uh, which will posted on the Q&A group. Ma'am, could you please tell me where I can get these recordings of the class? Okay, let me paste this link in the chat box so you can have easy access. Please, you can go through all these recordings in my on my YouTube channel, which the link of this is pasted already here. So thank you so much for joining today's class. Next class will be on next Sunday, 4 to 6 p.m. And thank you so much for connecting with this session. And if you have any doubt, please you can stay and feel free to ask. Otherwise, you can leave the meeting. Thank you, Shehnaz. Thank you, Shivasami. Do you have any